Hello, everybody. So today, NICOLAS CAPTIER will make a talk on feature selection by Boruta and Lasso methods. Thank you very much, NICOLAS, that you agreed to make a talk. And please start.
Thank you, Alexander. So good afternoon, everybody. So today I will briefly talk about some feature selection methods. Let's say that I think this is an informal meeting, so maybe there will be some inaccuracies, but we can still talk about it afterwards. This is an introductory talk, meaning that I can give you some hints about these two methodologies, but of course I'm not an expert, so maybe you will need for further details to search by yourself. But it's just to give you some starts, let's say. So I will start this presentation by giving you like an application or an introductory example of why and in which setting we could apply such methodologies. And the introductory example is about gene regulatory network inference. And actually, this topic has led to the development of many methodologies and many statistical analyses. So as a disclaimer, I'm not a biologist, so it will be really basic. But let's say that within a cell, gene expression can be regulated by the expression of other genes. So you have target genes, let's say, and transcription factors that can either activate or repress the expression of genes through many different processes, very complex ones, direct or indirect one. This is not actually well known now, if I'm not wrong, but the idea for sure is to be able to for one target gene to detect the possible transcription factors. So this has many applications. We can think of drug developments. So maybe if we can act on some transcription factors on specific genes, maybe this can linked to new new therapeutical strategies. For sure, for system biologies to be able to build gene regulatory networks and to associate with one target gene the transcription factors is really important. So from all this occurs the idea that we would like to associate from one target genes all the different transcription factors. And so over the past years, many methodologies have been developed to reach such a goal, right? So you have an example of one article, let's say it's an arbitrary article, which in this article they try to infer from gene expression this kind of regulatory network. So for each gene, you want to associate all the genes that can regulate, either activate or inhibit its activity. And so the main idea of all these methods is I want to collect data for each gene, and then using this data, I want to infer association. So maybe one of the most popular data that is now collected for gene is, of course, gene expression data. And so the question that many people asked was, can we, from gene expression alone, infer some relationships and some transcription relationships for different genes, right? So you have many methodologies that have been tested, the most simple ones being computing some correlation between the different genes, and obviously more complicated methods like Bayesian network methodologies, and I will talk about this later, methodologies based on machine learning and feature selection. linked to this topic, actually, you have many well-known challenges. So the Dream 3, Dream 4, and Dream 5 challenges, and people worked on this question on inferring some gene regulatory networks from gene expression. So the Dream 3 and Dream 4 challenges are associated to simulated networks. And the Dream 5 was way more complicated because it's associated with in vivo networks. So this is just an example, and in the following, I will not claim that the method that I will discuss about are the best and for sure will help you solve such a problem. In fact, inferring gene expression, gene regulatory network is really complicated, and maybe this is an utopistic task to think that using only gene expression we will be able to infer such network, but because you have many challenges to overcome. The mechanisms associated to transcription regulation are really complicated, and for sure, the only gene expression does not carry all the necessary information. You want also maybe to have some to infer some causal relationships, so meaning distinguishing the direct links between genes from the indirect ones. And also an interesting question is, once you have inferred your relationships, how do you validate them? So for sure, this is still a very challenging task, but I think you have new opportunities now that will improve this task. Now, when you look at the literature, many people are trying to use multimodal data. So meaning not only gene expression, but all the kind of omics data to see whether it can improve the detection of relationships between genes. Of course, we can combine also in vitro experiments, so perturbation analysis, for instance. So I think a lot of work needs to be done and can still be done about this task. But this is not my main point. This was just an introductory example about one specific method that actually was partly developed to try to solve such a problem, and which is feature selection, but in the specific case or for which you have a lot of features. So let's say that you have a lot of genes, X1 till XP, for instance, and one target gene, right, which is Y. And the question that you ask yourself is, can I identify among all the genes the one that are the transcription factor associated to the gene Y, right? And people have used, let's say, one ideal hypothesis, meaning that in an ideal world, we can think that the expression of the transcription factors is sufficient to predict the expression of the target gene. And so, in fact, we go back to a problem which is a really not basic problem, but a problem which is really famous in machine learning is, I have a predictive task. I want to predict the expression of one gene, one target with all the variables. And my question is, how do I select the variable which are relevant for this task? And so here in this slide, I have two main problems, two main challenges associated to this problem. And in the following, we will see that the methods that I will present will try to address such challenges. The first one is that usually you will have a lot more features than a number of samples. And this is a statistical problem, and we need to use some algorithm that can deal with such a problem. And the second problem is a problem called all relevant selection problem. If you are familiar with machine learning, you have many methods to select features, but usually the aim of machine learning is not to select all the relevant features. It's to select the best subset of features that will help you predict the target. But it does not mean that this subset of features gather all the possible and all the relevant features. It only gathers a subgroup which is optimal for this algorithm and for predicting the target. But here the question is a bit different, is I want to identify all the different features, whether they are redundant or not, I don't care. I want to retrieve all the possible transcription factors. And I want to finish this introduction by giving you like the, let's say the basic way of doing so, and giving you the, let's say the main ideas behind feature selection in machine learning. So feature selection in machine learning, you have like 3 steps, and you will. I don't want to be too. You will find them in most of the algorithms. The idea is first you train an algorithm, right, to predict from all your predictors your target, which is here y, and then you use different kind of strategies to measure from this algorithm the importance, the impact of each variable. So you can rank at the end the different variable related to its importance in the predictive algorithm. And you use this ranking to select the one that are the most important. So the 2 methodologies I will discuss in the following will basically be based on this kind of concept. But for sure, they will try to add more complex strategies to address the 2 challenges I discussed in the previous slide about high number of covariates, and also try to detect as many as possible relevant features. Okay, so I will try to present the first method, which is about which gathers 2 concepts, which is the Lasso algorithm. We will see, but I'm sure that most of you are familiar about this algorithm, and this notion of stability selection. So this is the only mathematical formula, but the Lasso algorithm is just, let's say, a way of regularizing the classical linear model. So the problem with linear regression that you have here in your objective function here is that when you have many more features than number of samples, you cannot no longer apply this kind of strategies. And you need, in fact, to regularize, adding a penalty on the parameters. You need to really regularize. You have many different ways of regularizing a linear regression, but one way is making an assumption on the sparsity of the solution. So you will say, okay, I want a solution with a lot of 0 coefficient, meaning that I want to turn off many features. I want to predict at the end my target with a very small subset of features. And you can tune the level of sparsity that you want in your model, right, using this hyperparameters lambda. So usually when you use Lasso algorithm, this is a choice which is left for the user. And here you have a representation of a really simple task. Actually, I took a screenshot on scikit-learn documentation, and from right to left, we will increase the level of sparsity. So increase the lambda coefficient. Each color represents the different features. And you do see actually that when you increase the level of sparsity in your problem, you will select less and less features. And you can have a kind of intuition saying that the most important features are the ones that are turned off at the end, basically. So the feature selection strategies I will present is based on this idea. In fact, the Lasso algorithm is in itself a feature selection strategy, right? Because he will choose to turn off some features that he considers not to be important for the regression task. But you have many questions associated to this. The first one is, which value do I want for the lambda parameters, for instance? Or is my feature selection stable if I add some noise to my data? So you have many questions that we want to address, and that's why the method I will present was developed. So these are the two problems I just talked about. The first one is that actually feature selection with Lasso is very unstable. So for instance, if you deal with very highly correlated features, if you perform the Lasso 2 times, it will one time select feature A as predictive, and one time it will select feature B. So of course, when you want to select all the features, including all the correlated features, this is a problem. And the other problem is at the end, if you say, okay, I retrieve the features that are selected by the Lasso, you don't have any way of measuring the confidence that you have in the fact that this feature is relevant or not. And so a very simple idea which has been proposed, and actually which is not linked to just Lasso, but to any kind of feature selection strategies, is called stability selection. And the idea is just to perform the experiment again and again, but at each run of the experiment, you add, like you resample your data using Bootstrap, for example. So it's a simulate variation in your population, and you perform the experiment. So for one run of the experiment, you will select some variables, and you do this again and again. And so at the end, you can score each of your variable with the probability, the probability of being selected. So it's just the ratio of the number of times the feature has been selected over all your runs divided by the number of repeats. And this will be your score. And it has been shown empirically that using such a score, instead of just one run of Lasso, increase the performance of the feature selection and stabilized it in a way. And so here you have a slide which gives you an idea of how to combine Lasso and this notion of feature selection. And it's taken from this article, which I sincerely recommend, because I think it's really clear and it's really well developed, but I have no time to go through all the details, and I have not the expertise also to go through all the details, but it's really interesting. And and I think it's one of the first time this strategy of combining Lasso and stability selection was developed. And so what they tried to do is they had a task that they wanted to select all the genes among like 4,000 genes associated to the prediction of vitamin B2 in some bacteria organism, right? And so this is typically the task that could be solved by the strategies I'm presenting today. And so the idea is based on two main steps. The first one is that we don't want to select a specific lambda, a specific strength of regularization, and instead of doing that, we will do the experiment for every lambda, every possible lambda. So it's the path that I've just shown you 2 slides before. And then you do this notion of stability selection. So on the left here, I don't know whether you see my mouth, but you have the value of the coefficient for one Lasso experiment. So as you see, when you increase the sparsity constraint, more and more features are turned off. And on the right, you have the results of this notion of stability strategies but for every value of lambda. So meaning that one run of the experiment will consist of applying the lasso for every lambda. And then you do that again and again with bootstrapped sample of your data sets, original data sets. And so what you see at the end here is that for each feature, we display the probability of being selected, the one I presented before, depending on the strength of regularizations. And the red features are those that are consistently selected over the different lambda, and those that we can confidently consider as important for the regression task. So of course, there is a key element also is once I have this, they call it the stability plot. Once I have this stability path, the question is how do I build a strategy to say these red features are important and the other are not. So what they do, and I think we can discuss about it, and other people have proposed other strategies. They say, okay, I will consider my feature as stable and important if the maximum probability observed over all the different lambdas is greater than a given threshold. And this is one of the problems of such a method. But they discuss about it in the article is how do you set the threshold. So do you want to retrieve the variable with a probability higher than 0.6, 0.7? You have to decide at some point. But they discussed quite heavily about it in the article. The second article, which is linked to this kind of strategy, is an article which developed a method called Tigress. And the method is based, again, on this notion of stability selection and lasso algorithm. But I wanted to talk about it because, first of all, it is specifically designed for this problem of gene regulatory network inference. And so you see here the results they obtained on the different network of the Dream5 challenges. And you realize actually that they perform quite well for the simulated network. But as soon as you try different methods for in vivo networks, this is much more complicated and the results are much less impressive, you know. But this is kind of expected. And of course, it was done in 2012, and now many efforts have been made to tackle such a question using other data. But my point here is just to present you another strategies which again is based on stability selection. And one thing that changes compared to the previous slide is that they used another strategy. Once they have this stability path I've just showed you, they tried another strategy. They compute the area under the curve of each path for each feature, which is just the average probability score across all the regularization strength, and this gives you a score. And if you want, you can just keep this score, and this is what they do actually. They don't put any threshold because the AUC is computed against this score, but you can also put a threshold and say, I consider my gene as relevant if their score is over a given threshold. But then you have to ask yourself, what is a good threshold? And this is not an easy task. So to conclude on this part, I would say some of my thoughts, and again, it's just like personal thoughts, and I think at some point you will have to explore by yourself also this kind of methodologies. But the first thought is that, of course, many hyperparameters have to be fixed and have to be chosen by the users, and it's like an important drawbacks. But still, in the article, you will find some discussion about how to what are the best strategies to set such to set such hyperparameters. The advantage of also such methodologies compared to the one I will present after is that the mathematical concepts behind Lasso are quite well understood, and so you can have some theoretical guarantees, or you can have some also statistical analysis. And you will see in the article that you can perform some technical stuff because we understand quite well the Lasso algorithms. Of course, one huge drawback, and actually the next method will address this drawback, is that it only takes into account linear relationships. But another advantage is that it's quite simple to implement, and it's also easily extendable to. Here I tackled the regression settings, but in classification settings, you could use logistic regression with N1 penalty, and it will behave the same. I think it's the same. It's the same strategy. Okay, now I think I will address the last and the second method that I wanted to present you, and this is a method maybe you will be less familiar with, but the concept is really simple, and it's called the Boruta algorithm. So let me start by introducing you a very famous algorithm for this problem of gene regulatory network inference, and which is called the Gini-3 algorithm. So the Gini-3 algorithm, probably some of you have already heard about it, is just based on random forest. So the main advantage of random forest is that compared to linear regression, for instance, it can model quite easily complex and nonlinear relationships, and also it performed quite well in this setting of high number of variables versus a low number of samples, because also it's based on the averaging of weak learners, but also that for each decision tree you will select a subset of features, and this is a way of managing the high number of features. And the Gini-3 algorithm, what they do is that if I want to infer link relationships between each gene, for each gene, I will consider the gene as a target, and I will try to predict it with other genes. I apply a random forest algorithm. I use the default feature importance associated to random forest, and then I obtain a ranking, and this ranking is used to infer the gene regulatory network. So using random forest is quite interesting for feature selection problem, but the questions that are raised associated to such a strategy are how to avoid false positive, meaning that you could select some genes that just by luck are associated with your target gene, but in fact, if you add some noise to the data, you will see that it's not real relationships. So how can you deal with that? Also, the question is that you see at the end with Gini-3, you have a ranking, but again, you don't have any strategies to say, from this ranking, I can decide which are the relevant features and which are the features that are not relevant. So it's a bit the same problem than the methodology I presented with Lasso, when you have to, at some point, set a threshold. Here, you could just set a threshold, but again, it's kind of arbitrary. And we will see that Boruta tries to develop a clever way of selecting the relevant features. So here you have, without further ado, I can just present you the concept of Boruta, because it's really simple. And the main idea of Boruta is adding another actually level of randomness, using not only the features that you want to test, but let's say shadow features or random features. So how do you create these random features? You just take your normal features and you shuffle them so that they will no longer be associated to the target, right? And so you use this new data, so the concatenation of real features with shadow features to predict your target, right? So you perform your random forest, and at the end, you can retrieve the feature importance. And so you will consider that the features that are really relevant for the task are the features associated with an importance higher than all the shadow features. So in fact, you will consider that if a feature has an importance which is comparable to shadow features, it's probably noise. And so at this step, you can attribute for each feature above this threshold, like a +1, saying you have been selected at least once, and the other features you attribute 0. And so doing so and repeating this strategy again and again, for each feature, you build a statistics, because for each feature, you keep into memory the number of times it has been selected by the algorithm. And so this is this part which is kind of clever compared to the other algorithms. So at the end, a way of saying my feature is really important is that I will compare the number of times it has been selected with just a random binomial distribution. Meaning that if the feature is random, it will maybe be selected with probability 0.5. But if my features is really relevant, it will be selected a high number of times compared to this random distribution. So you can build a really simple statistical test with the hypothesis H0 being my feature is not relevant and follow a binomial distribution, and the hypothesis H1 is my feature is really relevant and I should keep it, right? And so this whole slide presents the Boruta algorithm. So it's based on, first of all, this notion of statistical tests, and also this notion of shadow features. So compare the real features with some random features. So I will follow with saying that for biological tasks, because this is what I'm interested in, and Alexander also, and I know that Marco is in the audience also. For biological tasks, the efficiency of Boruta algorithm has been shown empirically. So let me be clear here. It's difficult for this kind of methodologies to have like theoretical guarantees. I cannot say that this is, I cannot give you specific theoretical guarantees, but people have tried it and have demonstrated that it was quite efficient when tackling this problem of selecting all the relevant features associated to a specific predictive task, right? So you have an interesting article here that actually tried different methodologies to select omics variables associated to a predictive task, and they have shown that Boruta was quite efficient to solve such a task. So I recommend the reading of such an article. Also, and it's interesting, the original implementation of Boruta was in R, so you can use R package, but I did not try it, so I cannot say whether it's easy or not to use. But you have now also Python implementations that are easily that you can easily use, and sometimes it's easily linked to scikit-learn, so meaning that you can use some scikit-learn estimators within your Boruta strategy. So I'm using this one. So you have the GitHub repository here. And recently, doing some research for this presentation, I came across this repository which implements Boruta, but also it claims to implement also some more complex strategies, but based on this Boruta principle. So I did not try it, but from what I've read really rapidly, it seems quite interesting. And also because I know that in the audience, at least Alexander is quite a fan of Kaggle. So I found a Kaggle here, which talks about Boruta and some implementation on Python. So it's worth looking at it. So I will finish, and I think it's my last slide. I hope I was not too long. So again, it's just an introductory talk. I will finish by giving you some thoughts about this method. So the method, and when you read the article about Boruta, it was originally built around random forest and the default importance metric for random forest, which is a Gini impurity. But actually, when you look at the strategy, nothing prevents us for using other algorithms, for example, or other importance metrics. For instance, if we could perfectly change the algorithm, so random forest is quite efficient. For sure, it can deal with high number of features, nonlinear relationships. But I was thinking yesterday, actually, that for instance, if you have some data sets with missing data, we know that you can try rather XGBoost, for example, which by itself will deal with missing data, and maybe you can just apply the Boruta strategy, but with XGBoost instead of random forest. And the other idea I had a long time ago is, for sure, the default importance metric is interesting with random forest, but actually you have now access to other way of measuring the importance of machine learning algorithms, and especially one which is really trendy now is called SHAP, which is based on Shapley values, and really quickly Shapley values consist of, if you have a game of which associates different players, they will receive a reward, and the question that a Shapley value asks is how I redistribute this reward among the different players so that each player received a fair amount of the reward compared to the effort he has made in the coalition, if I am clear. But this is now applied to machine learning algorithm to explain machine learning, and you can perfectly think, okay, I will replace the default metric in Boruta by this SHAP values. And actually, a long time ago, and I implemented some strategies, some similar strategies in this GitHub repository. So to be honest, it has been a long time I've not used it or implemented. So it's for sure not perfect, and I'm sure that you can improve it a lot for the implementation. But still, you can have a look and see whether it could be interesting. It was particularly linked to inferring some association between features, which is exactly the subject of this talk. But I think it's worth looking at it and trying to improve or to clean the code, because it's not particularly well optimized. So I hope I was quite clear about it, and I hope I gave you some hints about this method and some motivations to go look by yourself and try by yourself. I do not claim that these methods are the best. I think they are interesting, especially this notion of stability selection for Lasso and this notion of Boruta also. And I think it's worth trying for different problems. Not only gene regulatory network inference, because this is really complicated, but other problems can be solved or can be tackled using this notion of feature selection and all relevant feature selection. So thank you for your attention, and I'm done, Alexander.
Okay, thank you very much, Nicolas. So colleagues, please ask questions or comments, suggestions, anyway. So now you can unmute yourself, or you can write a question in the chat.
Oh yeah, I saw some
Yes, there is a question in the chat. So the question is about transcription factors. So the question is that sometimes the expression of transcription factors themselves is quite low. And so is this the main problem dealing with transcription factors? Or the other problem, which is to connect them with the genes which we want to find as their targets?
I think both problem are matters actually. So yes, I agree that sometimes it's the transcription factor won't appear really clearly in the gene expression data themselves, but also there is a problem about which methodologies to apply. Yeah, I think one of the main limitation of inferring transcription factors from gene expression alone is that you don't have enough information. I think we know that you have very complex mechanisms between behind the regulation of genes, and it's not necessarily at the level of the RNA that you will be able to see that. So again, I'm not a biologist, but from what I read, I think this is really complicated.
Thank you very much. Any other questions or suggestions? Please unmute yourself. Boris, do you want to ask a question?
Yeah, I wanted to ask a question. Thank you, Alexander. Thank you for a very clear talk, Nicolas. It was instructive. I have a question. I'm not a specialist in analyzing biological data either, but just about the motivational part. Again, I haven't worked with ElasticNet extensively. I've heard that in biology, it's often the case that you have sets of features which are interdependent with each other, like gene sets, basically, which would, I assume, random genes from those gene sets would be arbitrarily selected by Lasso on different runs of the algorithm. So they're basically the same. And I've heard of ElasticNet approach for handling such groups of features. Did you work with it? Can you tell anything about it?
Yeah, I think the difference between just Lasso and ElasticNet is that ElasticNet you add L2 penalty, and basically, if I'm not wrong, the effects of adding an L2 penalty is that instead of just removing one of the highly correlated features, it will lower both coefficient for both features. So in the case in which you want to select all the different genes, I think it won't solve the problem, because at some point, maybe you will when you will analyze the coefficients at the end of your ElasticNet, if you have a huge set of highly correlated genes, you will see for each of them really small coefficients, but because they are highly correlated, if that makes sense. So that's why this notion of stability selection tries to counteract this effect, saying, okay, let us take the Lasso, but I think it's the same with ElasticNet. If I redo the experience again and again, maybe the gene A, which is really correlated with the gene B, will be selected 50% of the time, and the gene B will be selected 50% of the time. And if I put a threshold, like I want all the genes selected more than 50% of the time of all my experiments, maybe I can hope that I can retrieve also highly correlated features. This is not perfect for sure. It's difficult to deal with highly correlated features, I agree.
How do you think, what would be the desirable behavior of a feature selector in such case? You would want to group all the genes in the gene set into a single feature and then select this gene set together?
Yeah, here I don't speak about necessarily feature selection, but when you want to apply machine learning strategies using genes, usually you would like to deal with highly correlated genes by grouping them together. So you can have several strategies. Maybe the most simple one is the one you are talking about, meaning that you can try to create a score for each group of genes. So one feature for each group of genes, and then use them as an input to your algorithm. So this could be a way. So for instance, you can use prior knowledge. So you know that some genes behave coherently. And so you can say, okay, I take the average expression of all the genes. This is the most naive approach. For sure, we can think of more complicated approach. Another topic is also using this notion of group lasso. I don't know if you're familiar about it, but so the penalty is no longer on each gene separately, but on groups of genes. So you can put as input all the genes and try to enforce sparsity, but at the gene subgroups level, if I'm making myself clear. So yeah, you have several strategies to try to deal with it, but this is a huge problem in biological data for sure.
But this is not what you're interested in with this approach.
Oh. Yeah.
Why doesn't it fit your needs?
I mean, for this specific task of saying I want to infer all the genes that are associated to a transcription factor, I want to be able to detect also the highly correlated genes, right? So I think if you aggregate, to be honest, I don't know. I have to check in the literature whether it has been done or not. But my intuition is that you have to take into account all the genes separately. But maybe you're right. Maybe aggregating them will reduce the noise and will help finding some. But let me give you an example. If your goal, for instance, is to say, okay, I want to find all the genes that are associated to target genes because my goal is to develop a new treatment strategies, meaning that if I find a gene which is highly associated to another gene, I will try to indirectly target the target gene by, in fact, targeting this transcription factor. So in a way, some people are really trying to have gene relationships. And another example is sometimes you want to infer this kind of gene regulatory networks to be able in system biology to develop some methodologies. So I'm not expert, but I think aggregating genes together is not a good option in every case. But for predictive purpose, I think you're right. I think it depends on what you want to obtain.
Okay, thank you very much. Let me ask also a question about the Boruta. So how many times should we make this simulation? I mean, repeating this.
That's a good question. I think this is an hyperparameter. I will say a naive answer. The higher number of times, the better. But this is really a naive answer. Maybe some works have been done. I have to check. Sometimes you can check the default parameters of the Python implementation or the Boruta. Sometimes it's really arbitrary, but sometimes they have tried different ones and they have concluded that this one was sufficient. I think this number of times is really linked to a trade-off between the computational complexity, right? Because for sure it takes some time to run, and the precision you want to obtain. But it will be difficult for me to give you an idea. I don't know, to be honest.
Okay, thank you very much. Any other questions or comments or suggestions? Okay, so if
no, maybe.
okay,
Okay, thank you very much for this very Curie representation, and maybe a little bit personal question, but from your experience, if you, for example, did a lot of computation of this kind, how do you feel the sensitivity of these methods? How can you, from your experience, from your data, what is the results?
From my experience is that for sure, if I take the Boruta algorithm compared to, let's say, more less refined strategies, you avoid many false positives. So actually, it's a good thing with Boruta that you empirically you avoid selecting some genes that are not relevant, but for sure not all of them. And to be honest, when I applied it to gene regulatory network inference, but maybe because the task was too complicated, for sure it did a lot of mistakes. But I don't think that it's necessarily linked to the strategy in itself. It's more linked to the task. For sure, when you apply it to real data, it's always complicated. So to be honest, it's difficult to. I think it's really task dependent also. I mean, it depends on the data. If the data if the relationships are quite clear within the data, I think you won't have any problem. If the data are really noisy, or if the relationships are really difficult to detect, I mean, I think any kind of strategies will have some problems. So that's why I put this. I put one review on biological data. They tried it and they were quite happy with it compared to other methods. But it does not mean that if you want to apply on other tasks, it does not mean that. I think one of the main advantage with Boruta is that it's again this clever way of deciding how to set the threshold using this notion of statistical test. But again, maybe I was not clear about this, but there is still hidden hyperparameters behind this, because behind any statistical test, you have this notion of p-value, for instance. So once you have done your statistical test, what p-value do you set as the threshold, meaning I consider the variable as relevant or not, is a bit hidden also. I think it's more clever than just putting an arbitrary threshold on the feature importance, but still you will have some tuning to do. And I think it's really trial and error. There is no theoretical guarantee, unfortunately, to my knowledge.
Thank you very, very much.
Okay, thank you very much. So let me also ask a question. So what you mentioned, I think it's not about single cell data. And now when the single cell data appears, so we have many cells, maybe hundreds of thousands. So are there any works or modifications of these approaches when we deal with such a larger data set? So the computational costs will be much bigger and so on. And
I'm
a little bit different.
I'm not expert in this, but I've seen some recent article that we are trying to use single cell data to infer transcription factor target genes relationships. So I think that to be honest, the methods I presented you, and especially the first methodologies like linked to Lasso, are quite old now. So they were developed like 10 or 15 years ago. So 10 years ago, I think there was a trend for gene regulatory network inference. It was with the DREAM challenges, it was the thing to address. And maybe after some years, people started to realize that it was a really difficult task. But now I feel like from the really short literature review I've done for this presentation, I've seen some new article trying to tackle this task and using some new data, so like single cell data, but also proteomics data. So I think with new data coming up now, maybe you will have like rebirth of methodologies to infer gene regulatory network. Because for sure, it's still a really interesting task for biologists. I think I've talked about example for developing new drugs, but also better know the biology about some specific biological mechanisms. So I think the rebirth is really based on this new data and how to integrate these different data. But I'm not I don't know any methodology to do that. But I think for sure this is an interesting direction to explore.
Okay, thank you very much. Any other questions, comments, suggestions? Okay, so if no more questions, thank you very much, Nikolai. It was very nice, clear talk. So thank you, everybody for hearing. Let us stop this meeting, and the recording will be on YouTube several days later. Okay, thank you very much.
Okay, thank you for listening to me. Thank you all.
Thank you.